This is Dr. Jabali, and I'm going to be talking about stem cells. Stem cells can be located on topic 1 in the IB biology program. There are a couple of understandings that you need to know about stem cells. One of them is we're going to focus on specialized tissues, how they can develop into being differentiated into a certain type of organism. We're going to understand what tissue are, we're going to understand what differentiation is, and how they become a specific function. We're also going to understand how differentiation happens depending on what kind of genes are being expressed, the relationship between differentiation and the gene. Moreover, another understanding we're going to figure out is what are the properties of stem cells. Like they're able to divide and they're able to differentiate. And depending on their locations, they have a specific type of journey that will become a certain thing. Moreover, we're also going to see how stem cells are so good in a sense that they're helping with a lot of diseases. So they're being used as a therapeutic use. So before we do that, we also have to know what are stem cells and how are they developed, where they're coming from, which areas of the fetus or the embryos are we picking up, which ones are the best, and of course, understanding different parts of the cells when they're going from being totipotent to multipotent to pluripotent. We're going to learn all these stuff in the next part of the video. Moreover, there is an application. There's actually two applications that it comes to stem cells. One, you have to understand how stem cells are used with the Stargardt disease. We're going to understand what is this disease, what is it caused, how it's treated, and another named condition. In this case, I'm going to use leukemia, which is a type of cancer. We're going to understand how stem cells are used with leukemia. Beside that, we're also going to talk about the ethics when it comes to using stem cells. If they're coming from the embryo or they're coming from adult stem cells. And what's the difference between them and which one is good, which is bad, which are the pros, what are the cons? We're going to look on overall all of these. So before we can start understanding how stem cells are used in disease and stuff, first we need to see where exactly does the stem cells come from. It all starts with the sperm from the father and the egg from the mom. When they combine together, these two, you eventually have a cell that is fertilized. And this fertilized cell becomes a zygote. So a fertilized egg is the same thing as a zygote. It's when the sperms and the eggs come together. So right here, you have the first thing. This is the first cell, which was the combining of the sperm and the egg. It becomes a zygote. Then you can see that it's splitting into two, and then it's going to split into many cells. And then eventually it will turn into a blastocyst, which is this one right here. Now, this is a blastocyst. It's like a lot of cells. By then, you probably already have 32 cells or more. And then they start shifting, and they're starting to have a hollow area here. And then they keep moving, and eventually they start folding. And as you can see, you're starting to see the head and the legs, and you have the umbilical cord, and it becomes an embryo. And then from there, it continues developing, and you can start seeing more clearly the heads and the legs and the body. So why am I showing you this transition? Because you have to figure out within this area where is the stem cells located. But before we get there, you need to know the difference between an embryo and a fetus. See, we keep calling it an embryo before the 8th week. So from the moment the sperm and the eggs get together, when there's fertilization, we're talking about zygote. From the moment it is a zygote, all the way to the 8th week, we keep saying embryo and embryo and embryo. After the 8th week, which ironically, the 8th week is when the heart has the first beat, to the day that it's born, we call it fetus. So fetus is 8th week on, embryo is 8th week before. Just to keep that in mind. So when we say embryonic stem cells, we are talking about the first 8 weeks. Adult stem cells is when they're babies. So clearly, anytime I say the word embryonic stem cells, I'm talking about the first 8 weeks once the sperm and the eggs get together. So we're talking during the embryo time. We're taking stem cells from the embryo. So going back to this, you have the zygote. Now the zygote, as you can see, there's a word here called totipotent, and we're going to understand more about the totipotent as we go along. But just to start a little bit, the word potent here is power. And toti means total, so it's total power. And pluri means many power. So we're going to learn more about what it means by toti, pluri, multipotent. But so far, We have the zygote, which again was the sperm and the egg, and then go through a lot of divisions and then become a morula. And then from morula they become a blastocyst. So these are just different names. That means the more cells they divide to a certain level amount of cells, they become a new name. So if I have to go back all the way here, you can see that this more likely to be the morula. So when it's just one cell, it's a zygote. Divides to 2, 4, 8, 16, 32, and it's going from morula to become a blastocyst. And you can see the blastocyst, you start to see a hole in between. While morula, you still don't. You just see a bunch of cells. But moving on. So we have, when we say the word oocyte, which you're probably going to learn this if you do IBHL, oocyte is the same thing as an egg. So when the egg and the sperm get together, you eventually go through zygote, multiply many times, and you become a morula, which in this case, it's totipotent. We're going to get to that too. Continue dividing into many more cells, you become a blastocyst. And now you're becoming a fetus. And remember, I said to the 8th week, it's an embryo. After the 8th week, it's a fetus. Eventually, after a fetus, the baby's out, normal. But when do we take the stem cells from? We take it right here when it's still an embryo, when it's still a blastocyst. So when it's still a blastocyst, we take these cells out, and these cells are the stem cells. These stem cells can become heart, nerve, immune, any part of the body. So there are decisions to decide what kind of functions they're going to be. It's going to happen here at the blastocyst time. This is where they define their destiny. Because after the 8th week, they already have their destiny to go. So if you want to get a stem cell, it's usually on the first 8 weeks. Now, let's understand what stem cell is. Now, here is one cell. This is a stem cell. Stem cell has two properties, and you need to know what are these two. One, it can replicate. That means it can do mitosis easily. It can duplicate easily. The second thing is that it can differentiate. Differentiate means it can get a certain function. It's like in the old days, there was this thing called virgin CDs. When I say virgin CDs, it's a CD that hasn't been, no music recorded to it. So it's a completely brand new, or let's put it this way, it's a folder. It's an empty folder. It's completely brand new. That folder can become any kind of folder. It can be a folder about soccer. It can be a folder about volleyball. It can be a folder about fruits. It's a folder that is completely brand new, but it doesn't yet have a function. Once that folder becomes, let's say, a folder about the heart, that's it. That folder will always be about the heart. Now, what's the difference? The folder that has not become anything, that's the stem cell. Now, the folder that has become a heart, that's a folder that has been differentiated. That means now you gave that folder a specific function. So, one is that it can duplicate. So stem cells can duplicate themselves. And the second one is that it can differentiate. That means it can become any function. It can become a heart, it can become a lung, it can become a liver, it can become a bone. That's the two properties of a stem cell. It can become anything it wants, and it can duplicate as much as you want. And because of those two important things, stem cells are very important in the medicine world. And we're going to see why. So, self-renewal through division and differentiation. Those are the two properties of stem cells. Remember that. So, remember how I was talking about totipotent and pluripotent? Here is what I'm talking about. If you notice, toti, it means total. And when I say total, it means they can become anything. Now, when I say embryonic, you remember again, that is the first 8 weeks. And that means any time from the moment it was a zygote to a blastocyst. Like here, you can see the zygote to a blastocyst, right? At this point, they are totipotent. So the zygote, any cells like the zygote or any cells that are in the morula, they can become anything they want. However, once they reach the blastocyst moment right here, they are not totipotent. They become pluripotent. And you're like, wait, pluri is many, but it's not total. It means they still can do many different types of job. They can become many different, but now they have a certain area. For example, It depends where that cell was located. So we have 3 lines of cell layers. We have the endoderm, the mesoderm, and the ectoderm line. Once you have located yourself in one of these 3 positions, now you are not a cell that can become anything. Now you're limited to a certain type of thing, but not one type. So if you, for example, if you become an ectoderm layer cell, that means you can become, for instance, a skin, a neuron. That's your destiny. You still can be different types of cells, but in the area related to the ectoderm. If you are a cell that is located on the mesoderm line, that means you can become a heart cell, a red blood cell, whatever it is. And if you are a cell that is located on the endoderm line, that means you can become the pancreas or the lung. So you still have many options, as you can see, but it kind of reduced. So for example, if you're a cell that is located on the endoderm line, you cannot become a skin, not anymore, because you're in that position. Since you are on the endoderm line position, you can only be something like a lung and pancreas. You cannot be the skin. So that's why you become many, not totally. So erase this to make it clear again. So that's the difference. Now you're going to say, what about with the difference between pluri and multipotent cells? So the pluri, that means you still have a lot of more options. That means you can become anything related to the area that you're working with. But when it's multipotent, you can be different types of lung cells or different types of pancreas cells. While endoderm, you can be the lung or the pancreas. But again, don't think there's just one type of lung cell. There's 2 different types of lung cells and pancreas. There's so many different types of pancreas cells. So if you are multipotent stem cells of the pancreas, you can become any different type of pancreas cells. But pluripotent means, you can become a pancreas or you can become a lung. That's what it means. If you're toti, that means you can become anything in the body. So just let's erase here everything and be clear. So in this area, we still have to figure out which part is an embryo. All of these can be part of the embryo, but usually the embryo is more this part right here, the totipotent and the pluripotent. This is more like the embryonic stem cell. That means the first 8 week. Now, you still can find stem cells in adults. That means when they already have developed most of the things. And these guys, you can find it places such as the bone marrow, the skin. These are basically more adult stem cell. But you'll see the difference. See, when it comes to embryonic, they can become anything. Adult stem cells can only become what the stem cells is coming from. For example, if you take a stem cell from the skin of an adult, again, if you take a stem cell from the skin of an adult, that stem cell can only be part of the skin area because it is a multipotent stem cell. If you take a stem cell, let's say from the bone marrow, it can become a stem cell of a bone. Again, it's just a multi. However, if you take a stem cell from the embryonic case, that you have more options. That's a little bit of a difference between the embryonic and the adult, is that embryonic, you can become anything. But if you take a stem cell from adult, you can only make a stem cell from the area that it came. For example, stem cell from the nose, probably just from the nose. If a stem cell just from the skin, then you can make other stem cells of the skin. That's the difference between those 2 types of tissues. Now, we just re-emphasize again on the idea. So if you're going from the zygote all the way to morula, they're totipotent. That means they can become any kind of cell. After you get to the position of a blastocyst, which is this area, these guys have become pluripotent. That means according to what of the 3 layers. And I'm going to explain more about those 3 layers soon. And then if you got to the embryo, which is around the 8th week above, going to the fetus, then it becomes multipotent. That means it's just in a certain area. So for example, if you're just a heart area, that means you can only become a certain type of heart cell. So let's talk about this right here when we talk about the pluripotent. Now, if you look at this, they usually have 3 layers. So I'm going to highlight this so you can understand. Let's say this is the layer on the outside. then you have the layer on the inside, and then you have the middle layers. So you can see that there's 3 layers. The top layer here, we call it the ectoderm, right? And then the middle one, we call it the mesoderm. And when I say the word derm means layer, skin, whatever you want. Ecto, this one means out. So ecto means out, meso means in the middle, and then we have the endoderm, which endo means in. So you can see ecto out, meso middle, ecto in. Now, if we go back and look at those 3 layers, right? You can see if the cell end up in the middle of these divisions, the cell end up on the outside. That means that cell end up on the outer layer, which is the ectoderm. So what is this cell most likely to become? This cell, because it was located on the ectoderm, right? It was located on this layer. That means its chance is that it will become the skin or the nerve. However, let's say that the cell, while it's going to division, end up in the middle one, the mirror area. So you end up in the meso. So if you end up in the middle, that means it's going to become or a heart cell or a red blood cell. And if you end up in the middle, which is the yellow, which is the endoderm, then most likely this cell is going to become or the lungs or the pancreas. So it depends on the position of that cell. It will tell you what is it going to most likely to become. So very important that you understand that cells, they usually are reproducing and growing. This is what happens. You go from being one cell to become a trillions of cells. That's because your cells are reproducing, making more cells, and eventually they're growing. And now the question is, how come some cells are becoming heart and some cells are becoming lung and some cells are becoming bone? Because some genes are being influenced by it, by the environment. And I'm going to explain this a little bit more in the next slide. So we know that genes influence cells to do something, but we also know that genes are also being influenced by the environment. So depending on the environment, they will trigger a certain gene, and the gene will trigger a certain cell to be something. So you can see here, these are stem cells. That means they're coming from the embryonic section. That means the first 8 weeks. And depending on their location, a mesoderm, an endoderm, or ectoderm, they will become a specific type of cell. Now, why is these locations so important? Because according to these environments, they trigger a specific type of gene. Let me explain how this works. So let's draw here. The cell goes through, so here is a fertilized egg, right? This is after the sperm and the egg join. So we have a zygote. Now the zygote goes through so much division. And it goes through so much division, but then it reached and the cells start dividing and then it start making formation. Then you have this huge formation. Basically, some cells end up on the outside layer of this baby. And then, of course, on the inside, you have another deep amount of cells going on here, right? And then let's use another color. Let's use green. And then you have cells that are in the middle here. So what happened? Depending on the position, or should I, instead of saying the word position here, we can always say depending on their environment they are. So are you in the environment of the outside? Are you in the environment of the middle of the cell? Or are you in the environment in the inner part, the inner stuff? So if you remember from what we did here, the outside is the ectoderm. The middle one is the mesoderm, and you can see the word M, middle, meso. And then the endoderm, which is the inside. So these are the 3 layers. These are 3 environments. So if you are on the outside environment, which is the ecto, the outside will influence the gene, and that gene will make you become a skin. See what happens? The ecto environment caused the gene to express skin. But let's say the same cell here was actually on the middle one, the meso, okay? The meso will influence the same gene, and that will cause it to become what? The heart cells. But then, what if that same cell, or the same gene, is influenced by the endo, the inner one, the inner layer? Most likely it's going to become what? the lung. So it is all this gene right here in the middle. Let's say this is the gene. The gene will express according to what the environment is telling to. So if the gene is located on the outside here, that means the outside environment will tell the gene become a skin. If the gene is located on the cells that are in the middle, then the mesoderm will say, hey, become a heart. If the gene is located on the inner one, the endoderm, it will tell them to become a lung. And remember, you do have that gene in all these cells, the in, the outside, whatever. But, and depending on their location, they will express differently. This is how it works. So, just to be clear, your gene expressed according to the environment. There's no such thing as a gene for the heart. There's no such a thing as a gene for the lungs. There is a gene, and that gene can be expressed in so many different options. But the gene is waiting for the environment to tell him what to become. So again, if the gene is on the outside, then it will tell him to be a skin. If the gene is in the middle, it will become a heart. If the gene is on the inner part, then it's the lung. So this is how genes express and differentiate. Besides that, we also have to go back to the idea of differentiation. So let's put here, what does differentiation mean? So right here, you have one cell. And this cell is just a stem cell, right? It's a stem cell. Does he have a job? No, it's just a cell. However, it can differentiate. What does differentiation means? Differentiate means I am gaining a function. I am getting a job. I'm determining what I am. If it goes to the environment A, its function will be swim. I don't know. Let's just give you ideas. If it goes to the environment B, its function will be to walk. If the cell goes to the environment C, maybe its function is to jump. So these are different functions. Differentiate means you are gaining a specific job to do. So as long as you're a stem cell, you can become anything you want. But once you differentiate into becoming a jump, if that's your job, you are forever jumping. That's what differentiation means. I finally got a job and I'm sticking to this job for the rest of my life. This usually is when you get to the adult cell, when you finally get a function, and that's your function for the rest of your life. Okay? So then we get to two types of stem cells. There is the stem cells that are found in embryonic. That means the first 8 weeks of your life. And then there's the adult stem cells. Can be anytime from your baby to when you're adult. Okay? So embryonic stem cells, these guys can become anything. They're still undifferentiated. I'm just going to write like this. It means they have not got a function. They can be anything. When it comes to adult stem cells, that's not always true. Most of the time, they're already being differentiated. So for example, if you have a skin stem cell, they already know that this cell will only create other skin cells. It will not create a heart cell. So it's more defined. This, for example, you can find in all the 3 layers, right? The ecto, the meso, the endo. These ones, usually, they don't find everywhere. See, you don't have adult stem cells everywhere in your body. That's not true. You can only find some some adult stem cells in your body. Places like the skeletal muscle, the bone, the liver, the skin. These are some areas that can find stem cells. But when you're an adult, the chances of you finding stem cells in adult is very little compared when you're having in a fetus an embryo for the first 8 weeks. That means the entire first 8 weeks, all the cells are stem cells. But afterwards, once they differentiate, that's it. There are not all of them stem cells. Now, this one, the embryo, they usually are multipotent, right? They are basically can become anything. And then here, they can only differentiate to a certain type. Like I said, a stem cell can only become a skin. Now, there's less studies done with embryonic, and here's the reason why. So if I go back here, all the way here, if I decide to take one of the cell and use it for studies, like I want to figure out how to change that stem cell into a heart, don't think that you just trying to do it once you will figure out. it's very hard. So, but when I took that one cell out, do you know that that part of the cell could have become the baby's leg? So that means you are killing the baby on the first 8 weeks. You cannot just take a cell. Any cell that you take from the embryo, you're already killing the baby. And remember, when you're taking that cell, let's say you took that cell, you don't know what is the communication. You still don't know what is the exact right kind of environment that you have to put it in order to become a certain thing. We don't know all the communications. We don't know how the genes are fully expressed yet. So when we take a stem cell from a baby that is not born yet, it doesn't mean that the first time you do an experiment, everything is guaranteed. No, we have to do millions and millions and millions of test trials to figure out what is the right communication to turn that cell into a heart. So basically, it's all about testing out. Imagine you take one stem cell from a baby, and then you have to go through zillions of trials, trying with different types of environments to make a heart. Don't think that they already know exactly what's the environment. They don't. They have no idea yet what exactly is the perfect environment to expose this stem cell to become a heart. Once they figure out what is the best environment that will cause this cell to become a heart, then that's not a problem. But until you get there, you are testing many baby stem cells. So that means you are killing many babies just by picking one stem cell from it. That's why, as it says here, there's not much studies on embryonic stem cells because this is very controversial. Just taking one stem cell out of the embryonic, you're already killing the baby. There's no chances. The baby will be born deformed. However, that's why we have so much more studies with the adult stem cells, because we can take a little bit of tissue of a nose of an adult and he's still fine. We can take a little bit of tissue of the bone and still fine. It's not like he's going to be deformed. So there's more studies with the adult stem cells and not with the embryonic stem cells. So if I had to put in a more quicker way about the difference between the adult and the stem cells, is that the stem cells can be anything. They can differentiate into anything. And these ones, they're limited. You can only be a specific type. You can only produce a specific type of stem cells. This one, it's that you kill the embryo, right? And this one, you don't kill the adult. Okay? Yeah, so this one, you can pick from anywhere and it still can work, which goes back to can be anything. This one is limited. You only pick a certain areas. This one, you can find anything, anywhere. It can be on the ectoderm, the mesoderm, esoderm, endoderm, ecto, whatever. This one, no, it's just a specific areas like the muscle, the bone, or the liver, not everywhere in the adults. So this one is easy to find, and this one is harder to find. Okay, and of course, this is controversial. It can be killing the baby. This we're not killing. So these are the differences between the embryonic stem cells and the adult stem cells. Here's another examples of what are the advantage and the advantage between the adult and the stem cell. Like I said, with adult stem cells, there is not much rejection. What do I mean by that? So for example, if the guy, let's say, he burned his skin and he wants to grow another skin. If he takes the stem cell of his skin on the nose and he make more of those skins and put it on his arm where he was burned, his body is not going to reject because it's his own stem cell. If it's his own, that means it's 100% match. There's less likely to rejections. But let's say I take my stem cell and I create the skin to give it to my sister. Most likely my sister's body is going to reject it because it might be just 95% match. There's that 5% that don't match. So my stem cells work on me. And my stem cells will not work that easily on somebody else. So there is a good side for that for the adults if I'm working on my own. And the embryonic stem cells is a little bit harder. Again, stem cells, your own stem cells work on you. Your stem cells working on others is very rare. Here, no moral problems, of course, because you're not killing the adult when you're taking their stem cells. But with the baby, you definitely are killing it. Now, the only problem with this is that adult stem cells, we can only do a certain type of stem cells. For example, skin cells will only make skin cells, bone cells will only make bone cells. Now, these guys, the problem here with embryonic is that we're killing the baby. So there's so many other advantage for these two, but as long as you're mentioning four things about the adult stem cells and 4 things about the embryonic stem cells, you're fine. Make sure out of those 4, 2 are for and 2 are against for each. So here's another examples of ethical concerns that it comes against or supporting it. Now, what's the support? You can change somebody's life, right? Let's say they have, like I said, they burn their skin and they need to replace that skin. The good news is you can take a skin stem cell and make a new skin and put it. Woohoo, you save somebody's life. The other thing is, for example, they can find cures for things such as diabetes and sickle cell. So some people have diabetes, they cannot produce insulin. But if they make cells that produce insulin, you can solve their issues. You can treat cancer. And you can also treat some other things that the embryo would actually be open to do anything, right? Because remember, the embryo can become anything. So these are the advantage for the research. Why do we want to do research? Because, again, working with embryo stem cells, we can manipulate them to become anything. And because they can help solve some cancer issues and they can cure some diseases. And it can help some people have a better life. What's the bad side about it? You're killing the embryo. There's no way you can take a stem cell and not kill the embryo. And people believe that the embryo has the right to live just as much as an adult. And of course, and they believe that as soon as the egg and the sperm are fertilized, that's it. That embryo is alive, and that means you're killing it. And they also say that not all experiments come out right. So there is a chance that you taking that stem cell, you kill that baby by taking the stem cell, and still that stem cell will not save lives. So there's this whole ethical that happens in different countries, and their government decides, are we doing the stem cell research, are we not? So some countries are completely against it, and some countries are actually doing it. So it depends really about the country. Now, since we're talking about using stem cells to solve diseases, there's 2 that you must know. One of them is the Stargardt disease, and we're going to see it. And the other one is choice, and which in this case I'm going to is leukemia. So we're going to talk about different, when I say therapeutic use, it means medicine use. That means how we're going to treat certain disease. The first one that you need to know is the Stargardt disease. This one, again, we are always using, in this case, we're using pluripotent stem cells. Again, pluri, in this case, it means that they are somehow powerful and they can change into many things. So let's start with the bone marrow first. The bone marrow first, it's usually there's these bone marrows. People don't know this, but it's not the heart that produces your blood. It is your bones. Your bones are live tissues and they actually produce blood. So your bones are the ones who are producing red blood cells, white blood cells, and platelets. It is your blood that produces, your bone that produces blood. And even more interesting is that your bone actually has stem cells. So if I take stem cells from the bone, I can even treat blood problems such as cancer with leukemia. So what is leukemia? For those who don't know what leukemia means, leukemia is when you don't have enough white cells. So somebody who has leukemia is a type of cancer that is destroying these guys, the white cells. So the white cells, they fight against infections. And when you are destroying these white cells, your immune system is dropping. And eventually the person has leukemia and they're about to die. But if you go to the bone and you take the stem cells out of the bone and you start producing a lot of, cells that are bone cells that eventually becomes blood cells, then you can go back to producing a lot of white blood cells, and that may solve the cancer issue. Sometimes we put radiation, which is chemotherapy, and when we do this, we're killing all types of cells, not just cancer cells. And that means the person is becoming weaker. But if we take the stem cells out of the bone and place it back, we can solve them. But here is the biggest issue, okay? Let's say you have the person right here. This person has leukemia. And this person is going through chemotherapy. Okay, so she has chemotherapy right here. Now, what is chemotherapy? It's radiation. Now, when you hit the chemotherapy radiation on the cells, let's say the purple here are the normal cell and the black here are the cancer cell. When you hit radiation here, the radiation is not just targeting the cancer cells, it will also target the good cells. That's why the person that does chemotherapy comes out weak and start to vomit all the time. And because they're vomiting every time, their body is getting weaker. But they're trying to destroy those cancer cells because those cancer cells are killing the white blood cells and the body's immune system is getting weaker. So the person is getting weaker. But they say, okay, since we did the chemotherapy, now the blood level has dropped. How can we replace that blood level? We can go back to get the bone replacement, right? Because we're destroying the bone. Since the bone has cancer, we're killing radiation, so the bone is becoming weaker. So bone replacement, how we can do this? We can get the stem cell from the bone. Now, whose stem cell are we getting? From this guy that's doing the radiation? No, because you're doing the radiation on this guy. This guy, his bone is weak. He cannot create his own things. So you need to find another person and take this person's stem cell from its bones and then place it back on that person, and then that person will build it up. But here is the biggest issue, okay? You're taking the stem cell from another person, and that stem cell you're injecting into that person. After, of course, the chemotherapy has destroyed so much of its bone. If that person doesn't have 99% in combination and match, that means match with the genes, match with the cells, with this person, his body is gonna do what? Reject all these stem cells that they used. So the best option is try to find your brother or your sister. Get the stem cell of your brother or your sister. Why? Because they're probably gonna have a genetic that is very, very, very similar to you. Then the chances of having no rejection is higher if you get it from your sister and your brother who have their genetic very close to you. But if you get in the stem cells of the bones of somebody that you don't know, more likely your body is going to say reject. It's a stranger. I don't know. Does not work. So leukemia can be very hard because even though you can get the stem cell from the bones of a sister or brother, sometimes your body will say, I don't know you, so I will reject you. So some people do cure from leukemia and some people don't. So it's really luck, by chance, that you find somebody in your family who has like a 99 or 100% match when it comes to the stem cells and genetic. So more likely twins are in a good term on this case. The second use of stem cells is the Stargardt disease. This one, every single IB student needs to know. First, you need to know what the disease is. The disease, it's a gene that is recessive and it's passed on from the parents to the child. What happens is this gene causes you not to produce enough vitamin A. And what it does is it destroys your retina cells. Retina cells are found in your eyes. So if the retina cells in your eyes are being destroyed, by the time you're 25, you become blind. You have to know these, by the way. Again, you have to know that it's recessive. You have to know that it doesn't produce vitamin A. You have to know that it's destroying the retina cells. And you have to know that it's becoming blind by the age of 25. Now, this is the problem with the disease. The good news is that you can go, we already know, we can take the embryonic stem cell and we can stimulate it to become a retina stem cell. And then we inject into the patient's retina, that means the eyes, and then the retina will build up and voila, you have repaired the retina and the person is back to normal vision. So this is one of the successful use of stem cells. Once again, you take an embryonic stem cell and you stimulate the stem cell to become a retina stem cells, and then you put into the patient, and then that retina stem cells start developing into a retina cell. And then the retina replaces the old one, and voila, you have a brand new retina, and the person is back to seeing clearly, and there's no blindness. So the visual performance of this patient is back to normal, most likely. So you have to know this very clear, and I'm going to circle again the things that you must write on an essay. So the things that you must mention that it's a gene that is positive, that is recessive. You must mention the lack of vitamin A production. You must mention that it's the destruction of the retina cells. You must mention that they lose vision, they become blind. Then you must mention the pathway to do it. And of course, the visual response. These stuff must be mentioned in your essay to get a full point for this case. And last but not least, there's two types of therapeutic use, which is the somatic nuclear cell transfer. These are more like lab stuff, manipulations. So if you look from the drawing here, you take an adult stem somatic cell. Somatic cell means it's just any cell in the body. And you take the nucleus out. That's what the word enucleated means. You take the nucleus out, right? And then you place the adult nucleus in here, and then you stimulate the production, and voila, you have an embryonic stem cell. So in this case, what you're doing is you're creating a completely new embryonic stem cells by getting rid of. So for example, again, you have an adult cell and you have a nucleus. This nucleus has already been differentiated. What do? Get rid of the nucleus. So now it's enucleated. The word enucleate means there's no nucleus. And from there, what do? You place it into an egg that has no thing, and you put a nucleus of the egg. Now voila, you go through many divisions, and look, you have now a bunch of stem cells, or should I say embryonic stem cells. Just to repeat, because some people will not understand. Here, this is an adult. An adult has a nucleus. This adult already has a specific job because the nucleus has been differentiated. What do I do first? Get rid of the adult nucleus. Then I have a stem cell here, an egg. And an egg has a nucleus. So I'll take the egg of the nucleus. You take the nucleus out of the egg and put it into that. And then you stimulate it to go through division. And voila, we are back into stem cells from the embryo. Why? Because we use the nucleus of a new egg. That's known as somatic cell nuclear transfer. You're transferring the nucleus. On this case, this is the most dangerous one, which what they're doing is they're adding factors, they're adding stimulations. It can be electricity, it can be some kind of gene, it can be a retrovirus, and they just throw these guys around the somatic cell trying to force it to become something, which means the word induce. The problem with this is that it can become a cancer cell, it can become a virus, it can become a bad gene. So trying to stimulate the genes or force the genes to express in a certain way can cause the cells to go cuckoo. So when I say nuclear reprogramming, that means you injecting certain types of material into the nucleus, forcing the nucleus to express differently. And that can cause the nucleus to go cuckoo, and then the cell go crazy and become ugly. It can become a retrovirus, it can become a transgene, it can be a cancer cell. That's what oncogenic means, it's a cancer cell. So this one is the least used by the scientists. They do not try to stimulate the nucleus to change itself. So that is all for when it comes to stem cells. We already know that tissues right here basically are made of cells. And of course, cells will come out to be specialized, and that means they're going through differentiation. And I know, remember, that you need to find the environment that will do the gene expression. And then from there, cause the differentiation. The zygotes, the first 8 weeks, right, with the embryo. Zygote is basically the sperm and the egg together. The embryo is the first 8 weeks. We know the two properties of the stem cells is that it can divide and differentiate. Most likely the stem cells that we're going to use are the embryo ones. Why? Because they can become anything. However, of course, there might be issues. You're going to talk about the difference between the embryonic and the adults. What is toti, multi, and pluri. Talk about the cause and symptom standards. We have done that. Explain how stem cells are used in Stargardt disease or leukemia as we did. And then where did you get the stem cells? We can get the stem cells again from the baby or from the adult stem cells. And what are the bad sides about this whole embryonic stem cells in adults, which we have gone through. And that's pretty much for stem cells. Thank you.